Hi friends, today we'll be talking about gene prediction methods in prokaryotes and eukaryotes by computational methods. The resources of entire discussion is taken from www.imtech.res.in. The objective of entire discussion is to find the genes and predict the genes through the computational methods. So before coming to this point, we need to understand that entire structures of the prokaryotes and eukaryotes genomes. As we know that eukaryotes genomes are more complex than the prokaryotes, and some fundamental knowledge also we must know, which we have discussed before, regarding that the promoter regions, starting codons, stop codons, ORF, entire methodology to find the proteins from the DNA via transcriptions and translations. So the data box, if we talk about here the data box, we are actually transcriptions initiate, and we know already that forward directions and reverse directions, even that some analogy about prokaryotes and the eukaryotes, and we know that operon concepts where in prokaryotes many genes are regulated by the one promoter regions, and in eukaryotes, which we say that each individual genes is regulated by the one promoters. So here we already know that what is open reading frames, entire methods, how DNA is transcribed into RNA by mRNAs, and translations we synthesize the proteins. So entire methods already we know that in terms of the open reading frames. So here we need to understand that the differences which are there in the prokaryotes and the eukaryotes, already the simple differences which we know that prokaryotes is not having nucleus, eukaryotes as having the nucleus. In prokaryotes everything is like entire ORF, entire process, transcription and translations process happening inside that cytosols, we say that, and where in eukaryotes all the transcriptions and translations initiate itself in the nucleus. And in prokaryotes, the advantage in the prokaryotes cell is like it's a simple gene structures, small genome size is there, so which ranges from 0.5 to 10 million base pairs, and no introns. Right now the beautiful the differences which we can say that in prokaryotes no introns are there, whereas in eukaryotes, right, the exons and introns are there. Exons which codes the particular amino acids or proteins, uh, by splicing the introns alternatively, right? And on this in prokaryotes, high coding densities are there, more than 90%, so you can say. And a part of this, some advantages are also there in prokaryotes, in terms of that, because of high density of the genes in prokaryotes, some genes overlap also, and some genes are quite short also, right? So there are many approaches are there, methods are there, which is used computationally to find the genes. One is called, the first one is the rule based, and second is the content based, third is the similarity based, and fourth is that pattern based, and fifth is that ab initio methods. So one by one we will be discussing about that now. Uh, all the methods which is used to identify the genes computationally. So first thing is the rule based methods. So rule based method is simple methods, right, and which is mainly applied in, to identify the prokaryotic genes. So rule based approach is only depends upon that start codon and stop codons in between all this ORF is happening, and ORF also combines all that start codons and that stop codons, right? So like if you say that start codon, um, where transcriptions uh, initiate, right, with help of uh, data binding factors, already you know that TATA, right? uh, so and it is stop at that stop codons. So this entire process which we can understand is called that the rule based, right? So now the content based, if we talk about the content based, so we will be discussing all these things in detail in later stage. Just simply I'm giving you the some brief about that content based, because many genes are there, many codons are there, right, and many promoter sites, we say that promoter sites, right? So promoter sites in prokaryotes cells, we say that promoters which are really regulate many genes in prokaryote in terms of the operon concept, right? And in the eukaryotes, which we say that all the promoter sites is made of to regulate each gene have their own promoter sites where transcriptions really initiate, right? So these all are that content based, right? And that similarity based, if you talk about that, okay, we have the gene sequence, we have the genes, we have to identify these genes based on the similarity. Search methods are there where we are identify some orthologous genes and paralogous genes. Orthologous genes, like the genes which have the same functions, right, and which actually comes from their ancestors, which we say that what do you say, mm, some process is called that, no? uh, like just from the ancestor, whereas the paralogous, which comes from that duplications approach, right? So and paralogous genes have the different functions. So these are the terms which we use to identify the similar genes which have the same functions. And here is pattern based approaches is also used to identify the genes based on the machine learning approaches, where we have to identify like, giving that uh, uh, a set of genes, right? A set of genes as an input, and then we will see that what are that useful genes are there, functional genes are there as an output. So these patterns we need to understand that. And the fifth approaches are that ab initio methods, where these methods is fully depends upon the thermodynamics properties, right? So like how the thermodynamics parameters which are responding or their genes behaviors, all right, in three-dimensional space. And of course for this force fields is required to understand the entire process. So if we talk about the simple rule-based gene finding methods, the rule-based gene finding methods, and definitely we are looking for that start codons, and then staying in the same frames, scan in the groups in three until the stop codons is found. So then if we add some approaches, some methods are there, some rules is defined for this, uh, that is called that if codons is greater than 50, assume it's a gene. And if suppose if number of codons less than 50, go again, it will go to that backward direction to the means start codons and increment by 1. Then we'll see that if start codon is there, then it will go to the next frame till that stop codons. So these are the regulatory methods are there. It will start from like suppose uh, C ATG is there. So once you will come like ACT, it's not start codon, then it's come to that CT CATG. If it's not a start codon, then it will come ATG. So it is the start codon. So always one increment will be there in the sequential levels to get that start codon to initiate the transcriptions. So increment by 1 and start again. So this is the concept is there. At the end of the chromosomes repeat process for that reverse complement methods. So content-based gene prediction methods generally, you know that RNA polymerase enzymes are there, which actually deals with that how translations going to code that with the help of mRNA going to code for a particular amino acids right here in the reverse direction data box, like just all the activities means controlled by that now RNA polymerase. And some sequences also we may get like a possible sequences like Shine Delgado sequences, which are we like assume that the regions of this type of sequences will be in forward directions where ribosome will bind to that initial protein translations, and due to that we may get that codon errors, biases, right? So in terms of like if suppose one um, gene code, right, codons like ATC, it doesn't code for any amino acids. So this is the kind of errors instead of like ATG, right, is a starting codons. And we may get like a high GC contents, right, GC contents in a repetitions way. So already GC contents like in which deals with the methylations of DNA in terms of that where gene activity will suppress actually, right, and it may like interrupt that amino acid synthesis, right, and is suppress that gene expressions, right, so interrupt the gene expressions. And now if we talk about the similarity based gene finding methods, so it talks about that like how we are going to find the similar genes from the databases and from the defined sequences in terms of the orthologous genes, paralogous genes, the genes which are having the same functions, genes which are having the different functions, right? Yeah, one thing which came to my mind now is that orthologous genes are actually that speciations events, right, is coming from the just species levels, and the paralogous genes are that based on the duplications effect, right? So, similarity based sequences all the genes from the related genomes and compare them by using that online tools and resources like already 1 and 2, you know, the blast tools where we are finding that similar type of sequences throughout the databases. And some disadvantages are also there by using these rules in terms of the orthologous and paralogous genes sometimes loses their functions and become pseudogenes. And not all the genes will always be known in that comparison genomes. And the base species of comparisons is not always obvious. So it's actually similarity search is a good practice to identify that similar type of genes, right? And different servers and different tools, softwares are there to analyze this type of things by using the similarity search methods. Now, if we talk about the machine learning techniques, how the machine learning techniques is used to identify the particular genes in terms of the ANN based method, hidden marker model, Bayes networks, these are the familiar method to identify the genes, right? Out of which today will be talking about ANN based methods. So entire ANN artificial neural network methods is based on only three layers. One is called that input layer, second was the hidden layer, and third one is that output layers. So input layer set of genes will be there which will be work as an input genes or input sequences like it will go through that input layers. Once this input goes to the hidden layers have some weight is defined weight, and this input will multiply with the hidden weight variables weightage, and then output it will go to that output layers. This output weight plus input weight multiply with the input, and this output will go to that output layers. So if this output it comes as expected, and it will go to the output layer. If this is called that feed forward methods. One method is called that feed forward method. But if this entire output, the input which is multiplied with the weightage which is there in the defined in that hidden layers, it doesn't comes as expected output, then it will go again to that first layer is called that input layer. This is called that like backward propagation method, right? So these type of methods which we use to identify, so some input will be there, input will multiply with that weightage which is there in the hidden layer, and then it will give the output as expected, right? So as expected means some functional gene will be there, some unique genes will be there, some EST will be there. It depends upon that how we are expecting, right? And the hidden marker models also is a random process methods where we are like identifying the particular type of genes based on the random process, right? So these and the Bayes networks are also there. These two things we will be discussing later stage, because these all are the computational methods which you may not understand at this stage, right, being as a biotechnology student. But I will try to make you understand, right, in later stage. And now ab initio methods, if we talk about that, no, ab initio methods. So here, as I told you, this ab initio methods is depend upon based on that thermodynamics properties, where we are like identifying some kind of frequencies which leads to identify the genes, all right, and we analyze some environmental parameters which are responsible to give a particular functional genes, right? So these are that different methods are there to identify that genes. Even that ab initio methods we use, that force field, right, and all the parameters in terms of that um, their bonding energy, free energy change, enthalpy change, right, and others environmental factors, right, and thermodynamics properties to identify that genes. So these are the also like unique methods are there, novel methods are there, which is used for this um, entire process. So already we know that analogy, we need to understand that how the promoter regions are there, transcriptions where initiate transcription region and then translations, and different introns are there in that eukaryote, and exons are there. Exons actually which codes for a particular proteins or amino acids, right? And the entire already exons are there, exons translate into the proteins. So these are the structures of the prokaryotes genome, sorry, eukaryotes genomes. So eukaryotes are more complex structures, gene structures. Large genome size are there, which is ranges from 0.1 to 3 billion bases. Exons and introns are interrupted because it's like alternative effects. Exon introns are there. Exons only code for amino acids. Introns will splice, right? So these all the things are there. So and low coding densities are there. Low coding density because of the introns less than 30%. We also understand that the possibility or density of this in terms of the humans, 3% fungus, it's a 25% and 60% in east. So because of that, alternate splicing effects for all the genes, right? So and considerable number of pseudo genes are more due to these effects. So finding eukaryotic genes computationally, like a rule-based, the similar approaches which is used for the prokaryotes, we will be using for eukaryotes also, right? But some content-based methods which we need to take care about that in eukaryote genomes, in terms of the CPG islands, where C and G contents are more in the sequences which suppress the gene expressions or gene activities, right? And the feature based methods also has been included in finding the eukaryotes genes in terms of their promoter site, donor sites, acceptor sites, and polyadenyl sites, which is there at the three ends, AAA signals. And similarity based methods, many methods are there, the same methods which is we used in prokaryotes to identify the prokaryotes genes. So there are many programs and softwares are there to identify the genes for prokaryotes and the eukaryotes for by using different methods which we have discussed, like a rule-based method. Gene finders are there. Neural network-based programs like Grail EXPs are there to identify a particular gene based on the hidden Markov models, genome scans are there. And all these things you have to try by yourself. The only for the things you must understand that is that for all the programs, input will be the sequences, right? And you have to explore these tools, these programs, right? If you feel any problems, you can approach me and you can ask in classroom itself, or whenever you find time, you can contact me, you can text me, you can WhatsApp me, you can mail me, and you can discuss in that Google Classroom domain also, right? So whatever the feasibility will be there for you, you can come and discuss with me, because if I say like no one has given any demonstrations how to use the WhatsApp, Facebook, Gmails. We have tried our own and we have learned these things. So like programs, the same things are there. Only the thing is you have to think logically and you have to click on the programs to get the output. See, right? So these the programs you have actually use it. Any difficulties you can approach me. So combined methods are also there, where and their resources URL also I have mentioned here. So please use these programs, and if you feel any difficulties you can approach me anytimes, right? And is one of the familiar tools which is maintained by one of my known scientist, and he is very familiar at national and the international levels, and Dr. Raghva from IMTech. He has like made one tools which are really used by us, and it's giving more accuracy, like this EGPRAD, prediction of eukaryotes gene. So E stand for eukaryote, G stand for gene, eukaryote gene predictions. So this is very familiar tools. You can use it, right? I even myself have used plenty times, right? So these the tools, the similarity search methods. You have to do first blast X against the ref sequence databases. Second blast X, you have to again do that blast for the first blast, and then their result detection will be the significance exons from the blast X output, and then blast in against the introns to filter the exons. You have to do it, and the predictions uh, you have to use by using the ab initio programs. And this actually EGPRED programs is based on the ab initio methods, right? And one more add-ons is there in this program is called the NN splice site, used to identify the splice site and compute the splice site for the introns and combined methods.